Hi, my name is Jason Gill, and I'm a member of the Center for Phage Technology at Texas A&M University. In this video, we're going to talk about the CPT's Galaxy and Apollo platform for whole phage genome annotation and analysis. The functional annotation workflow. The CPT's phage genome annotation process divides the annotation into two major steps. First, structural annotation will predict the locations of protein-coding genes, tRNAs, and transcriptional terminators. The functional workflow analyzes the protein-coding genes identified during structural annotation to determine their function by a combination of similarity and conserved domain searches. In this talk, we're just going to be talking about the second part of this process, which is functional annotation of protein-coding genes. To begin the functional workflow, we will assume that you've already completed structural workflow and you have already identified all the protein-coding genes. The functional part of the workflow will predict the functions of these proteins based on a combination of BLAST, conserved domain searches, and tools including InterProScan, TMHMM, LipoP, and others. The CPT functional annotation workflow is our most complicated workflow. This is a schematic overview of it here. Obviously, we're not going to go into it in great detail. The functional predictions are going to be conducted on protein sequence. DNA sequence similarity degrades rapidly, and amino acid sequences are more constrained by the needs of maintaining protein function. This is why almost all protein functional prediction is based on analysis of protein sequence rather than DNA sequence. The CPT Galaxy Functional Annotation workflow executes 63 individual processes to analyze phage protein coding genes. In the end, it will produce 18 evidence tracks in Apollo, and these are grouped into 6 track groups, which will appear in the CPT Web Apollo platform. The CPT Galaxy functional workflow uses BLASTP and InterProScan as the major evidence sources for annotation. There are also multiple specialty analyses that are conducted as part of this workflow to identify things like phage spanins, lysis proteins, genes that are interrupted by introns or program translational frameshifts. The outputs of all those workflow components appear after the functional annotation workflow is completed. We're not going to deal with all of those in great detail in this talk. All the evidence tracks that are produced will be displayed in Apollo as shown below. So we're going to be talking mainly about BLASTP and InterProScan analysis, because these are the two main general tools that we use to predict protein function. The CPT Galaxy functional workflow uses BLASTP against 3 major databases, which appear as 3 separate evidence tracks in Apollo at the completion of the workflow. There is a custom database, which is comprised of RefSeq records from 37 well-studied canonical phages. Some of the most famous phages that are in this database are T4, T5, Lambda, T7, and so on. All of the phages which are in the canonical phage database have been selected because they have some historical significance and at least some level of literature support to support some of their annotations. The functional workflow will also analyze phage proteins against the SwissProt database, which is a high-quality protein database manually curated by EMBL and SIB. And finally, we run BLAST against the NR database, which is constrained only to bacterial viruses by tax ID. So keep in mind that this NR search is not against the entire NR protein database in NCBI. It's against only proteins that are found in any bacterial virus. So this is not a single tax ID constraint. It includes all bacterial viruses, including single-stranded DNA phages and RNA phages. The 3 BLAST results will appear as 3 tracks in the Web Apollo interface once the workflow is complete. The other major evidence source for functional annotation in our workflow is conserved domain searching using InterProScan. InterProScan is hosted by EMBL and is integrated into UniProt. InterPro integrates multiple conserved domain databases and assigns a single InterPro ID to related domains. InterProScan is a tool that searches a protein sequence against the member databases and detects conserved domains. This is an example of BLAST results in Apollo. The user annotation is in the yellow user-created annotation track at the top. In this case, we have the BLAST NR results in orange below it. Like other evidence tracks, BLAST results will be mapped back to the phage genome and aligned under each gene. The length of that alignment is visualized by the length of the bar below the gene. If the subject sequences are longer than your gene, they will be truncated so they don't overlap with other genes and distort the display. However, if the alignment is shorter, they'll be truncated appropriately. The E-value of your match is roughly visualized by the intensity of the color in the bar underneath. If you mouse over a bar, that will preview the match as shown here, showing a blast hit to a protein identified as a portal protein found in a Salmonella phage. Clicking on any one of these individual bars or blast hits will bring up the blast result window for that hit. This includes a lot of information on the quality of that particular hit. The score line is the blast E-value. The lower is better with E-values, and this is also reflected, as I said before, by shading the featured track. Blast results are automatically filtered to exclude all results with E-values of greater than 10 to the minus 3. The blast hit description is also provided. This may be informative and is entirely dependent on the quality of the annotation of the protein record that was used to make the database. This line is pulled directly from the protein entry in the database, so the quality of that description will depend on the quality of the protein record that it was deposited. Also, underlined in red at the top of the window, by the name, is the accession number of the hit protein, which could then be used to go to its record in the appropriate database. You may want to go into UniProt or NCBI to find the protein record and get more information on a given protein. Also of use is the alignment, which is in the gray box in the middle of the window. This shows the match line for the alignment between your sequence, which is the query, and the subject, which is the hit in the database. Obviously, the higher quality alignment here indicates a more reliable blast hit. This is an example of the canonical phage track output from the functional workflow. This is a custom database that contains only paradigm phage with some amount of associated literature. The database includes only 37 phages, so simply mousing over these hits can give a good idea of what type of phage you might have. For example, if you have a large number of hits against phage T4, this would suggest your phage is similar to T4. SwissProt contains the manually curated SwissProt database, and these protein annotations are generally quite reliable as they have all been manually reviewed by staff at SIB or UniProt. So this is an example of InterProScan results in Apollo. Again, the user-created annotations appear in the top, and the InterProScan results are in the white field below. InterProScan searches for conserved domains in 13 member databases. Not all of these domains have been integrated into the InterPro system. The hits here can be used to predict protein function based on conserved domains. It's important to be aware of things like domain swapping, especially if you have a hit to a relatively generic domain like an ATPase domain as part of a much larger protein. Conserved domain hits are more sparse than the BLAST results and often only align to a portion of the protein. Multiple InterPro databases will often find the same or similar domains, but there might be slight variations in how they're named. Again, clicking on any one of the individual InterPro domain hits will bring up a properties window. Not all the databases that are members of InterPro are very feature-rich. Some of the more useful hits when examining this track in Apollo are usually PFAM and TigerFAM, which have more data contained in this properties window. If the domain hit is part of InterPro, there will be a dbxref entry which has an InterPro domain identifier. An easy way to get this is simply to highlight that text in the window with the mouse and right-click on it to search in Google. And that will then bring you to the InterPro entry at the InterPro website. You can then find out more information about the nature of that domain. The calculation of the score for conserved domain searches varies between the databases. This is not strictly an E-value, as you might see it in BLAST results. These tools are all pre-calibrated to show only the results that they think are significant, so you generally do not need to evaluate this value, as you might for a BLAST result. In making a protein annotation, you are generally going to be using either your BLASTP results or conserved domain results, or a combination of the two. When evaluating BLASTP results, it's important to think about what database your hit is in. The preference for databases generally is to use the Canonical database or SwissProt databases before moving on to annotations from the NR database, because the NR database has not been curated. You're relying on the skill of other annotators when pulling annotations from that database, and also keep in mind that annotations in GenBank are not generally updated as new literature becomes available. It's also important to think about things like what the E-value is, Does the alignment span most or all of the protein between the subject and the query? And also, does it make sense that a protein with that function to actually be in your phage? When evaluating conserved domain results, you should think about how specific or meaningful that domain function is. And again, does it make sense for your protein to have that function in your phage? It's also important to make sure that the conserved domain really means what you think it does. You should really go to the InterPro record for that domain before assigning a function to your protein based simply on the domain name. Annotating your phage proteins using BLAST and InterPro results, you're going to have a mixture of hypothetical proteins and annotatable proteins. Proteins that have no BLAST hits in any databases and have no conserved domain hits will be annotated as hypothetical proteins. If your protein only has BLAST hits to proteins of unknown function and no other conserved domains, this will also be annotated as a hypothetical protein. NCBI is no longer recommending a distinction between hypothetical novel and hypothetical conserved proteins because that status can change when the database is updated. If you have BLAST hits to proteins with annotated function that you believe, or conserved domain hits that you can interpret, then you can annotate that protein with the appropriate function. To add an annotation to a gene in Apollo, in the user-created annotation track, you can right-click on that feature and then click Open Annotation. You can also alt-click on the feature. This will open up the Annotations tab on the right side of your browser window. This tab will show the gene and the mRNA parts of the GFF3 gene models that underlie the Apollo record. You can click on the mRNA field, so the mRNA feature is now active in the Details tab below, as shown by the green arrows. If you enter your new annotation in the Name field, you can then click Sync Name with Gene to copy the annotation to both the gene and the mRNA fields. These tabs may take a few seconds to update, so you can click Refresh, or you can click on a different field or tab in the window to make it refresh. What you need to look for here is that the user-created annotations track at the top of the Apollo window, that underneath that gene, your new annotation should appear. If your annotation appears underneath the gene in the user annotations track, that means it's been saved in Apollo. Another feature here is that you can add comments so that you can write some notes to yourself that might support the evidence for your functional annotations. For example, you can have the evidence of the BLAST homology to a known protein's conserved domain, or the results of external analyses you might run, such as HHPred. You can go to the Comments tab and enter a comment in the box and press the little plus button to add it. You can add an unlimited number of comments to each individual gene. And when the genome is exported, these comments will be exported with the record. Before you submit your record to the public databases, though, you may wish to clear out these comments, particularly if they contain large numbers of personal notes. For more information on using the Galaxy platform, we encourage you to review the extensive Galaxy training resources that are available online. These include those available at the main Galaxy homepage, Galaxy 101, and of course the Galaxy training materials produced by the Center for Phage Technology, which are specific to our system and located at cpt.tamu.edu/training materials. The CPT training materials contain detailed instructions on how to upload and manage your genome in Galaxy, and how to run the major analysis workflows. If you use our system, we also encourage you to cite our publication in PLOS Computational Biology that describes the platform and the associated workflows. The CPT Galaxy Apollo platform is supported by the National Science Foundation, Texas A&M AgriLife Research, and Texas A&M University. We would like to thank the many programmers and scientists who have helped design and build the system, and the excellent and supportive Galaxy and Apollo user communities.